Hello, this is to see how can we culture the bacteria and observe under the microscope. So here what I am doing is I like to show you the how the bacteria would look like under the light microscope. So once you have cultured the bacteria using a culture medium, then if you like to observe the bacteria, so what we need to do is once the bacteria are ready on the slide, even this can be even if you have not culture but you have the permanent slides on the bacteria, both gram negative and gram positive, then still you can observe under the microscope. So using a virtual lab we can see the understand how the bacteria would look like in the culture medium. All right, this is what I'm going to show you. This in a way I'm just trying to show you how the virtual lab can be used to enhance your laboratory skill. Now just to show you some features how to use it, you will see on the top theory, procedures, self evaluation. There are questions that will help you to understand better, and these animations would help you to how to use these whole instruments. And simulations is the this is what I have opened right now. You can change different variables and different slides and then rotate and then try to observe how it looks like. All right, so this is the experimenting space. Then there are some tasks given, and there are also reference book where you want to refer about that particular bacteria or the anything that you like to read about the particular specimen. Is the this reference book is available here. And now if you look at this is one, this is light microscope, and there are variables that you need to change, and you can see how the bacteria look like under the light microscope. So variables number one, when you try to view the specimen, you have an you can use mono locular or the sorry, monolocular or bilocular, meaning you have one eyepiece or two eyepiece for observation. Okay, then we also have 10x, 40x and 100x magnification. Then selecting the sample, you can select a gram positive, gram positive, then there is also gram negative. So these are, and there is also bacillus and bacillus, yeah, you can see this bacillus bacteria, and there is a gram negative and gram positive. All right, then there is another space is for to increase these measurement magnification measurements. Maybe you like to see this would be just, you will see later on if you like to see or how you want to increase the magnified areas, maybe which area you like to see. So this is what you will have to do, magnifying again the measurement of each specimen. All right, let's go for that. I will just demo one. So in order to do this, you need to I think I have all the specimen ready. Then this is the microscope that we are going to use. So what we need is there is a reset the positions. Here you need to resetting position and then keeping everything ready. Now if you want to observe the microscope under the microscope, this is what you are going to see. If you like to see a particular area, maybe you need to go and then focus highlight sorry, point a particular area, and then you can see on the screen here. All right, so this is the screen, and this is the one which you see on the slide itself. Okay, so just look at that way. I have moved my this objective piece. If you click this, it will move. So it is quite interesting that it moves. So now you saw this is for 0, this is comes to 10x, So 10x you are going to see your specimen in this way. And if you move another one, this is what you are going to see is in the 40x. So you are going to look at this, and also you can change your magnification from here. See, it is moving automatically here. All right, this is what you will see. And if you like to move this object, probably you have to go to the you can move these which is this. I'm not sure with this small plus minus, but you can see these these slides. I'm trying to look at if there is any tool that I can use. This is giving a power. So you see this is making this off and this is on. So careful in selecting some of the tools you can use, and you need to be very careful while setting. So you can do this. You can also see the pointer will turn into finger when you reach to a certain area, that which means you will be allowed to change. Okay, you are allowed to change, move, and even here at this, if it points there, no, it's not pointing. Okay, and this one, okay, up and down, and this one is for, okay, can you see there, just above that it is moving towards and this is for moving. Look at on the stage, it is moving. So this is very important that you move up, down, okay, or move up. So you can do this to move your slides up and down. Okay, this is the area, you know how to use the computers. Okay, so this is one thing that you can record. And right now I'm using this, let's go for the bacillus cell observations under the microscope, and let's use, this is how it looks like under the 10x. Now if you go to 40x, it will look slightly different. See, this is what it looks like. Yeah, you can see it. This is how the bacillus bacteria looks like. And this is by you can also change this. This is called binoculars, where you can binocular can be used. And yeah, so when you use the binocular, you are using a light. So you need to move your slides. Okay, you are moving up, down. Okay, then also moving this direction. Just look at this way I am doing it. You can take it off, make it clear. Okay, better. So let's make it to 10x so that we start with the lowest one. See, this is what you will see it, and then let's bring it this to the center. Okay, I'm trying to bring it at the center. Okay, this is how you can do an experiment and try to see it, and make it to 40x. Still it goes other side. You can bring it in like this. Okay, and then bring it to the center. Let's see the another bacteria. Now we are not able to trace any. You can go to 10x, okay, then reset, then observe under the gram negative. You don't, okay, this is gram positive. Using a binocular, you can use monocular, and you see this is the bacterial. In fact, this is the bacteria that you see, but with the binocular you can also see the same specimen. See, these are the one, okay, bacteria that you see. This is 30x, So you can go to 100x. What happens? See, this is what you will see, and this result need to be recorded and then kept into your uh, books, and then show this is and bacteria, gram positive bacteria at what is this, at 100 magnifications. Okay, 100x. So what you do is you can snip these very well. You can use the snipping tools, and you can copy this particular result to your book, and then bring it like this and copy, and then you can take it to your notebook that you like to keep records. All right, maybe I have one kept here, and let's see. Yeah, this is the one. So I have already kept aims, theory, material required, procedures, observations, and I have also put some of my observation of the route. But for you, you don't need these. So what I'll do is I'll just remove this. I'll remove this. So under the observations, I'll just paste it, Control V. So this is what I saw under the 100x, right? Now you may also bring for 10x, 100x, and 40x, and then do a comparative, and then share your observations. Okay, and then share your observation. This is how you will maintain your practical record. This is simply, I wanted to help you how to use the virtual lab. I hope this will help you, because please try to follow all the links that I have given. Thank you.